Hello dear students, welcome to the video lecture series on Western blotting from Unit 4A Immunoblotting Techniques under the syllabus Pharmacy Council of India 3rd year B Pharmacy 6th semester subject Pharmaceutical Biotechnology, subject code is 605T. Myself Dr. J.P. Lavande, Associate Professor, Department of Pharmaceutics, Shivlingeshwar College of Pharmacy, Almala, Taluka Ausa, District Latur. In the Western blotting, we saw Western blotting diagram and the detailed protocol for the Western blotting. The protocol consists of the 9 different steps, out of which the first 3 steps, that is sample preparation, preparation of gel, loading the samples, we already saw into our previous video. Now in this video lecture we will go through the step 4 to step 9. So step 4 is running the gel, step 5 is gel staining, step 6 is transfer, step 7 is blocking, step 8 is antibody incubation, and step 9 is antibody detection through the process of color development. So just to have a recap of Western blotting diagram, it includes sample preparation, that is protein extraction, followed by protein assay, running it through the gel electrophoresis, staining the gel, transferring protein to the membrane, either nitrocellulose or PVDF, then staining, then blocking, washing, incubating with primary antibody, incubating with secondary antibody, and the detection under the chromogenic detection and the chemiluminescent detection. So step 4 is the running the gel. After loading samples, place anode and cathode of electrophoresis chamber cap appropriately, and electrophoresis is carried out under proper voltage. In discontinuous system, voltage for stacking gel 70 to 80 volt is lower than voltage for resolving gel 90 to 110 volt to ensure that proteins are concentrated on the same level before running into resolving gel. Run the gel, and when bromophenol blue dye molecule reaches the bottom of the gel, the power is turned off. This process usually takes 2 to 3 hours. Step 5 is gel staining. After electrophoresis, booster comessive blue staining solution, comessive blue fast staining solution, and protein silver staining reagents are applied for gel staining. This is to check if electrophoresis works. Stained gel cannot be involved in subsequent membrane transfer procedure. Step 6. Weight transfer. Step 6 include take out gel and immerse the gel in booster transfer buffer for 15 to 30 minutes. Based on a sandwich model, install electric transfer clip by stacking a sandwich of sponge or transfer pad or gel or nitrocellulose membrane transfer pad or sponge, which is clamped tightly. No air bubble within it should be there. Ensure the gel is closest to the cathode and membrane closest to the anode. Note: sponge, filter paper and nitrocellulose membrane should be immersed in transfer buffer thoroughly. Add transfer buffer to the top of electrotransfer tank and insert electric transfer clip in it. Place the tank at 20 degrees Celsius. Membrane should be closest to the anode. Negative charged amino acid and protein will migrate toward the anode. Concurrent current of 150 to 300 mA is usually applicable. Transfer time depends on gel concentration. Membrane staining is recommended to check transfer efficiency. Place nitrocellulose membrane in booster imprinted membrane fast reversible protein staining reagent for 5 to 10 minutes and visible red band will appear. The membrane may be destained completely by repeated washing in wash buffer. Step 7. Blocking. Rinse blotting membrane. Rinse blotting membrane 3 times using booster wash buffer at room temperature for 10 minutes. After rinsing, immerse the blotting membrane in booster blocking buffer. Incubate for 1.5 to 2 hours at 4 degrees Celsius under agitation. Step 8. Antibody incubation. After being blocked, the membrane is incubated with a primary antibody which binds to the target protein, and then incubated with enzyme conjugated secondary antibody, HRP conjugated antibody and AP conjugated secondary antibody. Dilute primary antibody in booster antibody dilution buffer. Incubate the primary antibody and the membrane at 4 degrees Celsius overnight. Wash the membrane with wash buffer for 3 times about 10 minutes each time. Dilute secondary antibody in booster antibody dilution buffer. Incubate the secondary antibody and the membrane at 4 degrees Celsius for 2 hours. Wash the membrane for 3 to 6 times, about 10 minutes each time. Step 9. Antibody detection through color development. The choice of Western blot antibody detection method depends on specific substances that are labeled or bound to the secondary antibody. ECL chemiluminescent detection system and DAB chromogenic detection system are typically used in Western blotting. Booster ECL chemiluminescent system depends on incubation of the Western blot with a substrate that will luminescence when exposed to HRP on the secondary antibody. The light is then detected and captured by photographic film. Step 9. Antibody detection or color development. ECL chemiluminescent solution. Add 1 ml water to 50 microliter color reagent A and 50 microliter color reagent B from booster hypersensitive Western blotting chemiluminescent substrate and mix well. Or add the same amount of color reagent A and color reagent B from the mix well. Cover the blotting membrane with colored reagent solution. Exposure. Record test result using autoradiography film or ChemiDoc in darkroom. Using autoradiography film, exposure time for the specific antigen depends on developing effect ranging from seconds to minutes. Using booster developing and fixing kit, wash the film in developing solution. Stop washing when the band appears and then resin the film in fixative. Note: Western blotting stripping buffer is recommended to remove primary and secondary antibodies on the membrane if proteins of the membrane needs to be reused. So dear students, thank you for watching. If you like this video, please hit the like button, share our videos to your friends and subscribe to our YouTube channel Dr. JPL Video Lectures by pressing the red color subscribe button to get notification about more such videos. Thank you.